Hello friends, welcome to BMH Learning. In this video, I'm going to talk about factors affecting stability of DNA. So you would be able to know what's that which is making the DNA stabilized. But first you need to know, what do we mean by DNA stability or a stabilized DNA? We know a double-stranded DNA looks like this, and two strands of the DNA are somehow connected to each other. Using some chemical or physical treatments, these two strands can be denatured or separated from each other via a process called denaturation. So the initial form of DNA prior to denaturation is actually the stable form of DNA. And the one which we are getting after denaturation is destabilized DNA. Now, to understand factors affecting DNA stability, first we should know what's that which is connecting these two strands. So, the DNA is made up of bases. We know there are 4 bases. Now, each base on one strand is connected to second base on the another strand with the help of hydrogen bond. This is the one thing which is broken during DNA denaturation. I mean the hydrogen bonds. Now concentrate. It's not only the hydrogen bonds which are making the DNA stable, but one more thing is here, and that's the stacking interactions. The stacking interactions are the one which are responsible for bases getting stacked upon each other. Now you must be able to name the factors responsible for stability of DNA. So these are the bonds which we just discussed. That is the hydrogen bonding. The hydrogen bonding in DNA structure is present in bases. But apart from this, the bases also hydrogen bond with surrounding water molecules. The heterocyclic bases of DNA have various groups like polar amido, amidino, guanidino, and carbonyl groups. These groups form a complex network of hydrogen bonds with the surrounding water molecules and the bases present on the other DNA strand. The second factor is stacking interactions. Interstrand hydrogen bonding is clearly important in driving the formation of DNA duplexes, but it is by no means the only contributing factor. The individual bases form strong stacking interactions which are major contributors to duplex stability. These base stacking interactions are much more prevalent in duplexes than in single strands. Base stacking interactions are hydrophobic and electrostatic in nature. These stacking forces depend on the aromaticity of the bases and their dipole moments. Base stacking interactions in nucleic acid duplexes are partly interstrand and partly intrastrand in nature. These interactions increase with increasing salt concentration. It's because high salt concentrations mask the destabilizing charge repulsion between the two negatively charged phosphodiester backbones. This was all. Thanks for watching.